All right. Thank you all for standing by and welcome to our webinar entitled Diverting Nutrients from Aquaculture Facilities into Additional Products. This webinar series called Freshwater Science will highlight Ohio Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal blooms and human decision-making, bringing applied research to the public on issues that face our Lake Erie communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today are Dr. Sylvia Newell from Michigan Sea Grant and Dr. Kevin Neves from Bowling Green State University. Dr. Newell is Michigan Sea Grant's director, a nutrient biogeochemist and microbial ecologist. Dr. Newell's research focuses on the effects of excess nutrients from fertilizer and wastewater on inland and coastal waters, particularly harmful algal blooms in Lake Erie. Her current collaborative work in the Lake Erie watershed focuses on engaging stakeholders to develop realistic pathways for nutrient reduction. Dr. Neves is a faculty member within the Department of Biological Sciences at Bowling Green State University. His lab is interested in the development of integrated multi-trophic aquaponic systems where the byproducts of one organism is used to grow other organisms. Such systems can be used to generate additional income while minimizing environmental impacts. His lab has also developed a novel system to reclaim nutrient-rich tile drain water from agricultural fields and utilize this to grow fish and plants. We're delighted to have both of them here to talk about their research today. Before we do that, just a few things about the presentation, the webinar itself. During their presentations, all participants will be in a listen-only mode. Afterwards, around 12:20, I will conduct a question and answer session. If you would like to ask a question during the presentation, feel free to pull up that chat feature at the bottom of your screen at any time during their talk, and I'll collect and pose your questions out to Dr. Newell and Dr. Neves at the end of their presentation. As a reminder, this webinar has auto captioning and is being recorded to be posted onto our website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. It will help us continue to bring you great webinars. So without any further delay, I'd like to introduce Dr. Newell and Dr. Neves presenting Diverting Nutrients from Aquaculture Facilities into Additional Products. All right. It is your floor.
All right. Thank you, Joe. Again, thank you to Ohio Sea Grant for allowing us to discuss our research. Sylvia and I have been working on this project for a while, probably far too long. And it's actually good to finally get a chance to talk about this project and share some of the results that we have. That being said, we're going to start by talking about, what is IMTA? It is this term that we're going to use a lot throughout this presentation. And basically, it's integrated multi-trophic aquaculture, or in the case of this project, it is multi-trophic aquaponics. And it sounds like it's a mouthful. It's basically just culturing multiple species of organisms at the same time. So whether it's land-based aquaculture, it's recirculating, it's in net pens off the coast, most aquaculture is monoculture, it's one species. And so by combining all of these different species together, we end up with this integrated system. And while I would love to take credit for developing this system, it's actually been around for quite a long time. There are multiple iterations that developed in Asia going back to about 2200 BC. And simply put, this was a system in which oftentimes carp were grown together in these ponds. And so we've got, a lot of things that unfortunately are familiar to us here in Ohio, things like silver carp, we've got common carp, black carp, big head carp, and then these prawns down here at the bottom. All of these different species of carp feed on different parts of the pond ecosystem. So there are some like brass carp that are feeding on vascular plants, silver carp that are feeding on phytoplankton. And these ponds were, we'll say, fertilized naturally to encourage that robust growth. Very simple, very easy to do, capable of producing a lot of products. This is what most people might think of when they think of IMTA, if you think about it at all. And in this case, this is a conceptual model put forth by Thierry Chopin. And we've got a net pen here in the middle full of typically salmon. And what we're doing is we're feeding these fish like you would any other monoculture of salmon. And traditionally, the waste would go out to the environment and might have detrimental impacts. But in this integrated system, what we have are a bunch of things like organic deposit feeders, things like sandworms that might feed on that uneaten food and waste. We've got filter feeders like mussels or clams or scallops that are going to filter out all the minute particles. We've got, again, some other deposit feeders here that might be feeding on the waste produced by the shellfish. And then surrounding this net pen, we typically would see things like kelp that are going to be extractive organisms, and they're going to be taking up those inorganic nutrients, the nitrogen and the phosphorus that are also released from these systems. So basically, when we get this balanced system with an IMTA, whether it's out in the ocean or whether it's land-based, we end up with a situation where we're not producing nutrient pollution. One of the side effects, if you will, of even a recirculating land-based aquaculture system is that there's nutrients that develop and they have to be dealt with one way or another. Working with some colleagues here in the School of Business, these systems oftentimes are more stable from an economic perspective. We have improved output. So looking back at that theoretical model, you can have scallops and sea urchins and kelp alongside with those salmon. Lower cost per operational unit. So you're still putting in the same amount of feed, but now you've got lower, those costs are spread out through more organisms. You get things like product diversification. So more things that can be sold. Risk reduction, right? If something goes wrong, it has to, you still have other products. And then one of the more novel things is this social acceptability, sort of this green labeling. People oftentimes feel better when they consume products from the system. So getting to the project, we've got our greenhouse here in the corner. This is on the north side of BGSU's campus. We've got this really old, antiquated, this is the best picture I could find of our greenhouse. And we developed, when I came here in 2016, we developed this large-scale demonstration system. And so we have 5-foot diameter tanks. And in these tanks, we had 2 of them full of yellow perch, and 1 of them full of crayfish or prawns. We initially tried freshwater prawns. Turns out not great because it's not the best environment for growing them. So we've done Orconectes rusticus, paper shell crayfish. And so the water flows from the perch into the crayfish, and then eventually flows into a 5-foot by 20-foot raceway before being collected into a sump and then pumped back to the fish. And as we were constructing this system, you can see here, you can see this raceway, this 5 by 20-foot raceway. You see the tanks over here on the left-hand side. We put these floating boards in the raceway. We drill holes. We support the plants in these little floating discs. We've got the sump over here on the right-hand side. We've got a pump. This is our chiller here. One of the problems with being in Ohio in the summer is it gets hot. And so we do need to chill the water. But aside from a sock filter in the sump, there's no other filtration on this system. So everything is done biologically. And then we've got our little seedlings here on the left-hand side. These are the 2 students who helped me to build the system, Lana Neff and Neil Collinet. And then we did this for a year and we kind of messed around. And I had a student who was working on an independent research project, and we just grew a whole bunch of plants in the system. And in brown, we have the growth of plants in soil. In orange, we have the growth of plants after 2 months in the aquaponic system. And there's really not much that can be taken from this. Nothing did really well, nothing did poorly. And then we did tomatoes. And the tomatoes in our system exploded. They just took off. After a few months, we ended up with this hot mess of tomatoes, and this looks terrible. I'm fully aware of that. Um, uh, Eventually, we had to develop a system. This is another one of the students that worked in the lab with me, where we had to support them and make sure that we kept them pruned. The tallest plant that we have to date is 14 feet tall, again, produced in this system. And again, we sort of fine-tuned our methods. And one of the really novel things about this system is that we started to here at BGSU look at some other aspects of the system. So initially, it was kind of a training system, if you will, demonstration for students to get hands-on experience. And then my colleague Jonathan Kershaw in Human Health Services in their food science department came to me about, do they taste good? Do the tomatoes taste good? Which sounds like a simple question. And so he had a master's student, Brittany Kralik, who would come in and harvest some tomatoes from our system. And this is a picture over here from our system. We harvested some fish from our system. And in the Human Health Services building, we ended up doing some blind taste trials. And so participants would come in, they'd be given tomatoes both from a soil-grown system in the same room and from our aquaponics system, two varietals. One was an early girl slicing, one was a cherry tomato, and they'd answer all sorts of questions. And what was really fun about this was that there really were no significant differences between the tomatoes grown in the soil, same variety, same room, same growing conditions, and the aquaponics. So here we have overall liking. This is our 1 to 9 scale, appearance, taste, texture. So between soil and aquaponics for the early girl, no major differences. From the cherry tomatoes, the soil maybe slightly better, but again, no significant differences. And it also carried over to the fish. So this was our aquaponic fish versus wild-caught Lake Erie yellow perch. And again, we've got the same 4 categories: liking, taste, texture, and appearance. And I would love to be able to say that the aquaponics fish are better. They have a higher number. It turns out it's not statistically significant. And then the last kind of little interesting bit about this was that when people were presented with packaging that had information about the products, it turns out it significantly improved their perceived tastiness, their perceived healthiness, and their purchase intent compared to either info absent, or if they were comparing it to, say, a wild-caught packaged fish versus an aquaponics caught fish. And this is all incredibly exciting. And I'm going to turn it over now to Sylvia, because one of the big questions that we had is, this is great, but there's so much room for improvement in terms of how we can scale this up.
And one of the big concerns in scaling it up is nitrogen, which is why Kevin and I ended up pairing up on this. I would like to point out that most of this work was done as part of the master's project for Kashan Gomez when I was at Wright State, and that he was a master's student at Wright State doing that. And so it is on the first slide, but I feel obligated to mention that and to give Kashan credit for the great work that he did. So one of the challenges with an IMTA, though, is maintaining dissolved nitrogen below toxic levels. This is an extremely basic version of the nitrogen cycle here. But the most important thing is that actually technically 4, if you included urea, which is not on here, 3 out of 4 species of nitrogen that are normally found as dissolved nitrogen in water can be toxic to fish at high levels. And so having enough water plants and really plants to take up the nutrients that are being put out by the fish and the prawns is really critical to be able to maintain the health of those fish. And so in this project, that's what we decided to focus on. So nitrate becomes toxic to fish at 1 milligram per liter, urea at it's like 30,000 milligrams per liter, which is much higher, and ammonia basically equally high. So nitrate was really the one of major concern that we were worried might build up and cause harm to the fish. We're also trying to figure out how to keep the nitrogen balanced. A, so it wouldn't build up and become toxic, but B, because the whole point is to have a self-sustaining, recirculating, contained system, and you want it to be, generally in balance. Okay, so in order to do that, we have to figure out sort of the the ratio of fish to plants. And that was one of our goals and objectives for this project. And one of the major important connectors here is that ammonium and urea are processed by bacteria called nitrifiers to make nitrate and nitrite. And they can stop just at nitrite, or they can go all the way to nitrate. And that's a problem because the nitrite is the toxic thing that can build up and hurt the fish. So we're trying to make sure that the fish in the biofilter are correctly sized so that the system doesn't become unbalanced with too few plants and too little nitrate, or that it's too many plants, but there isn't sufficient nitrate to get the tomatoes to grow. So we're trying to figure out what's the rate of nitrification and how does that then balance between the fish and the tomatoes. So our goals, as Kevin pointed out, were already to evaluate the feasibility of the system, which seemed to be working pretty well. And then we're trying to measure the rates of nitrogen cycling to optimize the growth and then try to figure out how many plants to fish do you need within the system for it to be successful and scalable. Because the idea is that there's a market in Ohio for aquaculture fish, particularly an aquaculture system that isn't contributing to our surface nutrient problem in surface water. And so we're trying to be able to figure out how can we scale this up if someone wanted to take it for a more commercial sized product. All right. So the only thing of that we did not do was we had a goal that we were trying to compare it to other systems, but they were largely outside the state. And this is a good time to point out that the entirety of this work was done during COVID at a time where only one person was allowed to be in the lab or in the greenhouse. And it was really, the logistics were kind of a nightmare. So it's really a miracle that we got the project done to begin with. Okay, so the grad student on this project, Kashan Gomez, made monthly trips up to BGSU, and he collected water and environmental parameter data. So the ambient nutrient concentrations, pH, temperature, etc. in each of the fish, I don't know what containers that you can see here, and the prawn container, and then at 2 points along the raceway, which is the thing that has the tomatoes in it. And he looked at all of those environmental parameters, plus then he took the water back to measure rates in. And so he was trying to measure rates of nitrogen cycling. So how fast does the ammonia become nitrate? That's the nitrification piece. So we use something called the nitrate pool dilution method that was originally developed by Carini et al. And then we looked at ammonium recycling rates. So how fast are the microbes basically taking all that fish poop and releasing ammonia from it? And how fast is the nitrate in the ammonia being taken up either by the plants or by microbes? And so we had these tracer addition experiments. This is literally Kashan's backyard because, again, COVID. And we did 15 nitrogen tracer additions for ammonia. And from that, we can figure out if you add 15 ammonia and then 24 hours later, some of it's gone, you can calculate the rate of ammonia uptake. And if we get new 14 ammonia in that time, this is a very simplified version of the methods, but in general, that indicates a release from, you can call it recycling, remineralization, processing, doesn't matter. And so he was able to show, these are the regeneration rates. So this is how much ammonia is being produced by the microbes. And you can see that this is for 6 months from June to November, and that over time, like at the beginning, the microbes are really active and then they sort of settle out into this nice, more steady state. The differences here in the two colors are blue and red. And I encourage you not to pay a whole lot of attention to the numbers because they are in, I just realized, micromoles per liter per hour instead of micrograms per liter per hour. But if you multiply it by 14, that would give you the number. And then we use a very similar method to look at nitrification, but instead of adding ammonia, we added 15 nitrate, and we looked at, again, how much of that was gone after 24 hours, and that told us the rate of nitrate uptake, and then how much new 14 nitrate was produced, and that gives us a rate of nitrification or the conversion of ammonia to nitrate. And this is really important because there's an intermediate step in nitrification that's nitrite, and that is what is most toxic at lowest levels to fish. And so this shows you the rate of nitrification here. Unfortunately, again, remember this was done during COVID, only one person could be in a lab at a time. And June and July did not work out, but that's okay because we had pretty consistent averages, even though the range is large across the different sites. The average value was actually very similar across all 4 months for nitrate production, so nitrification. And again, these are in micromoles per liter per hour, but if you multiply by 14, it's basically between, 14 and 28, 30 micrograms per liter per hour. So it's actually a pretty high rate of nitrification and higher than the rate of bacterial remineralization and production of ammonia. But that's not the only thing that produces ammonia and urea, right? You got fish pee to deal with, and we didn't get that in our bottles. I mean, the fish, we probably got fish pee. All right, so if you look at this in general, the really nice thing is that the total nitrogen in and out for the mass balance of our system came really close to steady state, which is great because it allowed the dissolved nitrogen species concentrations to all remain below levels that are toxic to fish. And I don't think nitrate's actually on here, but it becomes toxic at 1 milligram per liter, and we were like 0.03. So it maintained it pretty well. We had about 200-ish grams of nitrogen added to the system every month via fish food, and about 200-ish left the system was changed within the system in multiple pathways. We had nitrate that was assimilated by tomato plants at about 12 grams per month. And then we had ammonia production at about 4-ish grams per month. But what was crazy mostly is that those nitrifiers were so high that they could produce about 92 grams of nitrate across the system a month, which is really rather impressive. Often we get, when we're thinking about macro systems, we think about nitrification as being a rate limiting step, as something that doesn't happen all that fast because the bacteria and archaea are, it's a very bad way to make a living. But this system seems to be great in terms of they don't have a lot of phytoplankton to compete with. They get a steady supply of food. The pH is good. So, and pH is pretty critical to nitrification. It needs to be within a relatively narrow band. So, and in this case, the average pH ranged between 6.5 and 7.5. So it's perfect for nitrification. It worked out really well. The plants were very successful. They, like I said, took up about 12 grams of nitrogen per month, and Kevin showed you the pictures, 14 feet high of tomato plants is pretty high. The one thing that Kevin didn't talk about that I think is maybe important to note is that the prawn tank was less successful. How many prawns did we start out with, Kevin?
Like 60, 75.
And by the end, there was one really giant prawn. So if you're trying to grow one freshwater lobster, it's a great way to do it. Otherwise, it might not have been considered a successful product, but it did do its job in terms of being part of the ecosystem and essentially eating the fish poop and processing it down in a way that made it easier for the bacteria to break it down and then provide a serious amount of plant food. So in the time of this 6 months, which mind you is not the entire 2 years it takes the perch to come to maturity, but in that time, there was about 6,000 grams of perch growth, and about 50 grams of tomatoes, or kilograms, sorry, of tomatoes were successfully harvested. And that had an approximate market value of $468, but that doesn't include the perch because they were not growing yet. As I said, the dissolved urea and nitrate concentrations remained well below levels that are toxic to fish, which would have been 30,000 for urea and 1 for nitrite. And so in general, the nitrogen balance was really maintained well with these high rates of microbial activity. And I think that that's in large part due to the successful growth of the tomatoes. They really did a great job at taking up the nutrients, that they grew like crazy. They provided an extra cash crop, and they made the system viable. And so in scalability, we figured out that there should be at least 1 tomato plant per 10 fish plus 4 crayfish. or prawns. Honestly don't know how critical the prawns are though. And that's maybe a question for Kevin, considering they ate each other at the end. But it does seem like this should be scalable. You just have to have a longer raceway and to have more room for more tomato plants, since obviously they go a little wild.
So one of the things that Sylvia and I were discussing throughout this was sort of the feasibility of it. And one of the things that we did not take into consideration and would be very challenging would be the costs of such a system. So from a short-term perspective, you're right, we can increase, profitability by increasing the amount of plants to a certain range. The problem is that more plants require more space, and more space might require more equipment, which might require, more inputs, including trace metals for the growth. That is one thing that we have also come to terms with in terms of working with an old greenhouse. Some other things too is the idea of, right, this idea of existing space. So we didn't have to build a greenhouse. We didn't have to pay for heating or lighting. Mainly because a lot of those costs could be prohibitive. LED lights can go for thousands of dollars to properly light up a system this big. Looking at this old greenhouse built in the 1960s, we would have designed one a lot better. And so there are options, there are ways to go about this. One of the things is, as you're taking into consideration a system like this, right? What is the goal of your system? Is it fish or is it plants? Because if it's fish production, that might dictate different equipment than if you're more focused on the plant side of things. Cost, if you as a viewer of this webinar were interested in constructing a system, what are you looking to spend on this system? This is this other system that we've been working on designing at the agricultural incubator on the north side of town. And it is a very cheap system. It is one that can be pretty easily designed, but it's not going to have all the bells and whistles, and it won't look as pretty, right? Location is always a concern in terms of where you're going to put your systems. And then if you have a system, are you capable of sort of retrofitting in, a raceway? If you currently grow just fish, are you looking at a new construction? Because again, most of that is going to add to your cost. So what Sylvia and I think we've done really well is demonstrated the feasibility of this system from a production capacity and from a sort of chemical feasibility. It definitely works. I think there's more work that does need to be done in terms of other considerations. So that's what we've got for you, and happy to take questions.
I just wanted to add that it might be an opportunity if there's an existing greenhouse or an existing hydroponics system to add fish into something that's based on plants and, allow the system to be recirculating. So we have some questions already.
We do. Do you want me to start them up? And we'll
Sure.
get to as many as we can here?
Sure.
All right, perfect. Well, so one of the questions that we had in general, and Kevin, you touched on this towards the end of the presentation, was some of the hurdles, the costs of doing this from a small scale perspective. I guess in general, between the two of you, what would you say was your biggest hurdle with this project besides COVID?
That was the biggest one on my end.
Yeah, I mean, hurdle logistically, it's while we're trying to measure all of these things, it's trying to keep everything in check, so to speak. So, temperature, for example, it was summer, and normally there's students in and out of the greenhouse. So it was a lot of babysitting the fish, basically. That is one of the hurdles. Yeah.
Okay. And Sylvia, it would be COVID.
Yeah.
I think it would have been a
Teaching someone
completely
how to
different
do
project.
things without being there physically and having to like FaceTime was very challenging.
All right. Another question we had was, are there any seaweed species that could be adapted for freshwater systems like this as an additional commercial product?
That's totally a Kevin question.
I was going to defer I was going to punt it back to you. If we're talking about a true seaweed, I'm not aware of any for a freshwater system. That being said, I have worked on saltwater land-based IMTA systems, and things like ulva generally lend themselves pretty well to serving the role that the tomatoes did in this project. I also know that there's a lab out in California that is also currently working with saltwater stuff and seaweeds, sea urchins, and I want to say white sea bass, that would be comparable to this. But as far as a seaweed in a freshwater system, not aware.
All right. Another question that we had was, and this would, I think, come back to you, Sylvia. Did you have any structures or substrate for nitrifying, is that right?
Yep.
Bacteria growth,
No, we
or
didn't
just the
have.
walls of the tanks and plumbing?
So I can answer that question because of the
Oh,
design of the system. So in the prawn / crayfish tank, we had basically what's orange roadside netting. That provided a substrate for bacteria. We also did put some bioballs in our sump, so the little plastic media. But it was actually very grossly undersized for the volume of water in the system.
And we did not have anything like you're thinking of maybe for denitrifying bacteria. Um, because nitrifiers want oxygen anyway, we didn't need to have anything for like really thick biofilms for them. And a
all
lot of
right.
them are free living to begin with.
Okay. Another question we had was, did you check the concentration of other nutrients like phosphorus, calcium, and of micronutrients?
We have since started doing that. Unfortunately, water quality parameters, while we were measuring them during COVID, were basically pH, temperature, and ammonia. Basically, the three most important things for keeping fish alive. And a lot of that was just because we had no undergraduate help. Undergraduates were not allowed on campus at this point. The only people I saw during this time were Deshaun once a month and our greenhouse manager, and we had to stagger our schedules so that we weren't overlapping. So it was as much as I could do working every day during this. I remember how terrible that is, and this is bringing back all the memories. So
Yeah.
unfortunately, we do now, and we do measure phosphorus, we measure iron, we measure nitrate, ammonia, pH, and really those are the ones that we found to be the most important.
So to be fair, we did actually measure nitrate, ammonia, urea, and nitrate in addition to SRP. So we did measure SRP, and I think for the tomato plants, the report that we got back from the lab did include PNK, but we didn't measure it in the water. But
All right.
since they were grown to 14 feet high, I'm pretty sure they had everything they needed.
Another question was, did you, and we've gotten a couple of questions dealing with the actual system, so let me just go through a few of them. One of them was, did you add fresh water or waste dirty water? And was this on a continuous basis or in batches?
Okay, we added water just to replace what was evaporating and transpiration. We never do we shouldn't say never. We don't do water changes on the system. So everything that's added is what evaporates, and it's usually less than 5 gallons a day. So it's batch.
Okay. Another question that we got was, a couple people were asking whether or not this was considered a modified aquaponics system.
Yes, and that's why with the crayfish and in subsequent reiterations of this, we do the paper shell crayfish, and we have 50 to 100 of them at any given time, and they do a really good job of eating the solid fish waste. So it is a modified aquaponics system for sure.
Okay. Another question that we got was, how do you balance access to sunlight to the components that need light versus shading to the components that do better in dark and low light?
We put styrofoam covers over the fish and the crayfish with gaps along either side to provide some lighting as natural lighting would allow for. So I don't know if any of the pictures were really demonstrated showing it, but I think if I go back to, there was one that showed the covers on the tank kind of blocking out some of the light in those fish tanks.
Okay. One question that we got was dealing with when Kevin, you were talking about the tastiness of the
Mhm.
fish, and they were asking whether or not how how if the group that were tasting it, if they are a good representation of the average consumer.
So they go through a screener. As far as tastiness, the way that it was explained to me was that because they are tasting the wild caught and the aquaponics fish side by side, that you're looking for differences between the two, not necessarily do they like fish at all. Although again, they had the pre-screen questions. In terms of a representative demographic, it is on a college campus, so not entirely. That would be questions better suited for my colleague, Jonathan Kershaw,
Okay.
but it's standard practice.
Another question that a couple people were asking was, will it be okay to reach out to both of you if they have some additional questions about specifics on your research?
Absolutely. And if anyone was local, they could come and tour the system as well and see it for themselves. I mean, one of the best things that I see about this system is that like we want people to go out and do similar things to it. That was one of the goals.
All right, well, perfect. What I'll do is I will, if you're okay with this, I will add your email into the email I send all the participants with the survey link. So just in case they would like to reach out to you, they can.
Great.
I
There's one
think
more
that's
question from Heather, and I just wanted to say that I think it's a good idea, Heather. I recently had the opportunity to tour a bunch of similar-ish systems in Chicago, and there are a lot of places that are really trying to grow plants, and they just sort of add the fish in as a nutrient source. And then they get another product out of it, which is great. So I think the real question though is, what do you do for the fish food, and in terms of cost, how does that offset, the fertilizer you would have given the plants? So it's a thing to consider, but since you can then sell the fish separately, not that OSU is necessarily trying to do that, but if you were doing this for a, for a marketable product, you could.
all right. Well, thank you. We've gotten a couple of people asking whether or not this webinar will be recorded, and it is. So I will be sending that link over to everyone. I think that I think that was all the questions that we have. And I see that we're actually a little over, so this is perfect. I wanted to again thank Dr. Newell, Dr. Neves for their willingness to talk with us about their research. This has really been a great discussion. So we really appreciate your willingness. I also wanted to remind everyone that the survey URL for this webinar is in the chat feature. So please take a few minutes to fill that out. This webinar series is sponsored by Ohio Sea Grant and will continue on in January when Ohio State's John Lenhart will be talking about his microplastic removal from water treatment research. The registration link is in the chat. Again, thank you, Dr. Newell, Dr. Neves, for your participation and your willingness to present your research. This has been really fantastic. And all the participants on the call, thank you so much. We hope this was beneficial, and we hope that you'll join us again in an upcoming webinar. Thank you and have a great afternoon. Thanks again. We really appreciate your willingness.
Thanks for having us.
Thank
Thank
you.
you.